Started. Again, welcome to today's webinar, Drug Development, Ataxia Drug Discovery Pipeline. Please note that all attendees are in listen-only mode and that this webinar is being recorded. I'm Lori Sjogren, Community Program and Services Director for the National Ataxia Foundation, and I'll be assisting with today's webinar along with Patient and Research Services Director Sue Hagen and Communications Manager Stephanie Lucas. Questions for the webinar speakers can be typed in the Questions tab found in your control panel. We'll answer as many of your questions regarding this topic as time allows prior to the end time of today's session. So as your questions arise, please send those to us. And any unanswered questions or unrelated questions can be sent to naf@ataxia.org. We have two excellent presenters for today's webinar. First, we have Dr. Vikram Shakkottai, who is currently Associate Professor of Neurology, Molecular, and Integrative Physiology at the University of Michigan. He is also the Director of the Ataxia Clinic and the Associate Chair for Research for the Department of Neurology at the University of Michigan. He is the current co-director of the Clinical Research Consortium for the study of cerebellar ataxia. We will also hear from Dr. Susan Perlman, who has worked in ataxia for over 30 years. She serves on NAF's Medical Research Advisory Board as the Medical Director for NAF. Dr. Perlman is the Clinical Professor of Neurology and Director of the Ataxia Center at UCLA Medical Center in Los Angeles. And now I'll invite Dr. Shakkottai to start us off. Welcome, Dr. Shakkottai.
Thank you. Thank you for having me, Lori. I thank you all for being here. We are meeting under unusual circumstances, as you all already know, due to the current pandemic. What should have been a seminar at the annual Ataxia conference is now being done in this format. So I'm thankful that you can all make it and we can actually have this happen. I wanted to introduce what we will be talking about today. So Lori, if you want to advance the slide. So what I thought I would do is introduce the idea of how we get from a promising therapeutic agent all the way to human trials to an approved agent for the treatment of ataxia. So my portion of the seminar is somewhat generic in that I'm going to talk about why it's so difficult to find something that's effective for treatment and what your role is potentially as a participant in clinical trials or a patient seeking treatments and cures for ataxia, and what your role is in participating in both these trials and making treatment happen. I think I want to say ahead of saying anything else, we are very grateful to patients with ataxia for being willing to participate in potentially therapeutics that are still in development, because unless participation in clinical trials is essential in order for us to find treatments for these ataxias. So I'm going to start with kind of how we go from how do we discover an agent for the treatment of ataxia. And I think it's important to bring up the concept of model systems. So what is it that you're trying to do in terms of treatment? For example, we know that mechanisms of disease in the different inherited forms or sporadic forms of ataxia are quite different from each other. So it's important to understand what you're modeling in a system in order to come up with a therapeutic. If you can advance the slide, Lori. So there's various different model systems for ataxia. Some of these are cell lines, which are individual cells that may express a mutation that is seen in patients with ataxia. Obviously, this doesn't have the complexity of the brain or even a nerve cell, but this may tell you something about a specific protein that we think is abnormal and that needs to be reduced for the treatment of ataxia. The other model systems are human-derived reprogrammed neurons. So this is, I think, an exciting new development that's happened in the past 5 years, where skin cells from patients with ataxia can then be reprogrammed to convert them into neurons. I think there's a lot of enthusiasm about using these for the treatment, and I don't think we're there yet for using these reprogrammed neurons to put back into people, but I think this is an excellent model system to study disease. The third model system I would really want to emphasize is these animal models, and this is primarily a lot of our genetics of ataxia and a lot of understanding about how the cerebellum and the circuits work is derived from model systems, primarily mouse models. And one may think, what does a mouse tell you? But the organization of the brain and the cells in the brain that are responsible for control of movement are very similar in mouse as compared to humans. We don't yet know how therapeutics will translate from mouse models to human disease, but I think mouse models have been invaluable in understanding kind of larger network level changes in function that model human disease. And the last model system to mention is non-human primates. There are obviously ethical issues using non-human primates, but I think given metabolism and other things that are more close to humans than any other organism, I think non-human primates still have a role in safety studies prior to human trials. If you can go to the next slide, Lori. So the discovery of a promising agent to finally a clinical candidate is a long and relatively arduous process. There is considerable amount of time and considerable costs involved. So I'm just going to lay out a potential scheme for how a promising candidate can then go all the way to becoming a clinical candidate. And by a clinical candidate, which you see on the bottom right, I mean ready for a clinical trial. This is not necessarily something that's going to even work in humans. So we start first with collections of compounds over in the top left. There are these model systems that I told you about, usually starting with what are called high throughput or in vitro assays. These are usually cell-based assays looking at specific readouts. For example, in an inherited ataxia where there's too much of a toxic protein, are there compounds that reduce the levels of this protein in cells? And then there are secondary assays where you want to look at whether the reduction in this mutant protein, for example, was related to a specific effect, or if it was just making the cell sick. So if you make a cell really sick, it's not going to produce any protein, but that'll also kill the cell. So it's important to know whether this is due to toxicity or is it related to specific engagement of a target, for example. And this cycle has to be repeated multiple times, validated, and for you to ultimately get some lead compounds. And then the SAR refers to structure-activity relationships. So these lead compounds need to be optimized in terms of structure. We know that in the ataxias, we're dealing with something that needs to get into the brain. The brain is a very hard has multiple lines of protection to protect itself from invasion from anything that enters the blood. And obviously you don't want bacteria, viruses, and other things getting into the brain. So there's a very strong barrier for agents that are administered by mouth or by intravenously to actually get into the brain. So that it's important to structurally characterize these lead compounds and make sure that they are brain penetrant, for example. And then we once you make new compounds, then they have to be redesigned and they have to be synthesized and then go through this process of the screen to see if it's doing what it needs to do. And therefore, this process takes many years, as summarized in the next slide. Lori, if you can advance the slide. So getting a clinical candidate involves identifying a target, screening of the drug, optimization of the target engagement, which is the structure-activity relationship. And once you have a lead compound, you want to know whether it's safe, whether it's actually getting into the brain, and then obviously whether it's doing what it's supposed to do. If we can advance to the next slide. So this process, this is a summary of why it takes as long as it does. So that initial process of the identifying some lead compounds and doing a screen can take anywhere between 3 to 5 years. And some of the time is because you have to make sure that the assay that you develop gives you the information you need. It takes time to synthesize compounds, develop leads, and typically it takes between 5,000 or 10,000 compounds or even more to identify a compound that's worth pursuing. This then is followed by preclinical testing, which is looking for toxicity and to make sure that the compound is actually getting into the brain. This is done initially in mouse and rat models, and then ultimately non-human primates, and this can take an additional at least 1 to 2 years. This is followed by the actual clinical trials in which a lead compound or a lead molecule is then taken into humans. And I'm going to talk a little bit about phase 1, phase 2, and phase 3 trials. Some of you have participated in some of these trials, so it's worth knowing what each of these means. So phase 1 trial is a study in humans with one of these compounds, usually in healthy humans, really to look at whether people can tolerate this and that it doesn't have any acute toxicity. A phase 2 study does often involve patients with ataxia. And again, this may not establish effectiveness, but it tells you how this compound works in patients with ataxia, whether they can tolerate it and whether it's safe in that population. A phase 3 study is often what can be a registrational study where compounds that have passed through phase 1, phase 2 are actually tried in patients with ataxia to see if it has actual clinically meaningful benefit. This is then followed by regulatory approval through the FDA in the United States, and this can take some time depending on what the indication is. Lately, the FDA, at least for orphan indications such as ataxia, has moved relatively quickly for regulatory approval, and following introduction of a drug into the market, there's post-market surveillance because sometimes rare toxicity is not picked up in these clinical trials, and it's important to continue to monitor to make sure this is safe and in addition to being effective. If you can advance to the next slide. So what is the role of a participant in an ataxia in a trial? It's important to understand what the agent is intended to do. For example, an agent that is symptomatic addresses a mechanism that improves symptoms of disease. So practically in a ataxia trial, this may be a shorter trial with endpoints that are directed towards clinical measures of symptom severity. As opposed to a protective trial where in the case of let's say a mutant ataxia protein, this would slow progression. And in general, for ataxias are slowly progressive, so trials need to be longer in order to account for the natural history and the variability of decline. If you want to advance to the next slide. So I think as a participant in a clinical trial, it is really important to educate yourself about what to expect from this trial. I know we are all looking for a cure. We're all looking for something that can improve our symptoms, but I think it's important to be informed about what you're participating in, and not merely say, I want to help ataxia patients, so let me participate in this trial. It's important to know how likely that this trial is actually going to benefit you or other patients with ataxia. So I think it's important to know what is the evidence that an agent works, and this is important to ask your investigators as to how this agent was identified. Was it based on animal models? Was it human cells? What are the endpoints of a trial? So what should I expect from this trial? Should I improve? Should I expect my ataxia symptoms will improve, or do I expect slowing? So slowing is not always easily perceived in a short time frame. So how not to be disappointed immediately after taking a potential therapeutic agent. So the third important thing, and I think I can't de-emphasize this, is, are the risks known? For example, can I die in the trial? And can my symptoms actually get worse? Because we're talking here about an investigational agent. Next slide, please. And now we are entering a really exciting era in ataxia therapeutics. We have multiple different preclinical studies, multiple pharmaceutical companies that are now interested in ataxias, and therefore there may be more than one trial that you are potentially eligible to participate in. And some studies may restrict participation in other studies. So it's really important to make an informed decision as to which study is most appropriate. Some of this information is from the investigator who conducts this study, and therefore it's also your role to consider what the investigator's knowledge of the studies in ataxia is, and whether they are actually giving you a reasonable input into all the other ongoing studies so that you can make an informed choice. And this is why I think it does make sense to go to an ataxia center where there may be more familiarity with ongoing ataxia studies and investigators who are actually familiar with ataxia. If you can go to the next slide, please. So these are some ataxia centers. And I think the NAF is a really great resource to help you find ataxia centers and providers. I'm now going to turn over to Dr. Perlman to give you more specifics about ongoing publicly available studies for ataxia.
Thank you, Dr. Shakkottai, for the terrific introduction. And I know we're sad that we can't be face-to-face with everybody who's attending this webinar, but hopefully next year things will be more on the normal protocols. So the NAF has a wonderful website which keeps up to date with current research, current clinical trials, as well as informational things about the individual ataxias. So this information is available on the website clinicaltrials.gov. And it was also abstracted on the National Ataxia Foundation website, and I have the address at the top of the page. So these are current studies going on at the ataxia centers around the country and around North America. The Biohaven study of troriluzole, which is symptomatic and potentially protective of nerves for patients with, in this case, genetic spinocerebellar ataxia. Biohaven has another study going on for people with multiple system atrophy or multiple system ataxia, which is a non-genetic ataxia with a different drug, with a different approach. Cadent Pharma has a drug that could be helpful for symptoms of ataxia. They're planning to begin with genetic ataxia, but they are not yet recruiting, potentially in part because of delays from the COVID-19 issues that are going on now. Riluzole, which is available in the United States for Lou Gehrig's disease, is being looked at in France for spinocerebellar ataxia type 2 and in Italy for spinocerebellar ataxia type 7. In Korea, a drug that is already approved in Japan as symptomatic for benefits in a variety of ataxias is now being tested in a different country on the other side of the pond from Los Angeles, as it were. We have often talked about doing some kind of trial for this agent in the United States, but we have not yet moved ahead with that. Columbia University has a study looking at gait and exercise. There's a group in Italy that's working with transcranial direct current stimulation, electrical stimulation of the cerebellum through the skull. There's a couple of groups that are working with ways to measure ataxia with wearable technologies, things like Fitbit type devices that could be worn by an ataxia patient who's involved in a study, and it would reduce the number of visits they would have to make to a clinical trial site because the data from the wearable technology could be downloaded over the computer. There's 2 natural history studies going on. One is for the common spinocerebellar ataxias, ataxia type 1, 2, 3, 6, 7, 8, 10, and another one going on in Europe looking at the same ataxias, looking at people who are symptomatic and people who are gene positive but may not yet have symptoms. And at the NIH, there is a separate natural history study going on for spinocerebellar ataxia type 7. Natural history studies are important, even though they don't involve trying a new drug, because they set the foundation for what these ataxias look like, how they change over time, what biomarkers, things in blood or spinal fluid or brain MRI might be good markers for disease progression and also disease improvement. So all the data being obtained from the natural history studies is going to be used to design better clinical trials to get some of these drugs in the pipeline approved. Next slide, please. These are some of the other studies that are in the preclinical ataxia pipeline. So these companies have not yet brought their drugs into clinical trials with ataxia, but we know that the ataxias are next on their list. And you can go to their websites. This is not privileged information. And you can look up their pipelines on their individual websites and see how far along they are. Biogen has an agent that could be helpful in multiple system atrophy. Execure is working on a new gene-directed therapy for Friedreich's ataxia. Ionis and Wave Life Science are also using gene-directed therapy in Huntington's disease and are planning to move ahead to the spinocerebellar ataxias. Lacerta is using AAV vectors, which are adeno-associated virus vectors, where a virus is emptied out and used as a vehicle to bring something hopefully helpful into the brain. And their pipeline includes ataxia. PTC Therapeutics is looking at a broad variety of approaches to modify defective genes. And Unicure is also working on a viral approach to help block a bad gene, as it were. They're beginning with Huntington's disease and will hopefully move forward into the spinocerebellar ataxias. You would wonder why now these companies have an interest in rare diseases. The things that may have led to some of the time that Dr. Shakkottai indicated from the first bright idea to designing a trial that could be helpful are typically delays in developing a safe and targeted agent, but also delays in developing the technology that we need to design these drugs. Antisense ASO technology, which you can see is featured in a couple of the companies that we have looked at, is a relatively new technology. And I know we've all heard of CRISPR, which is an even newer technology, which is being considered for application in genetic disorders. 15, 20 years ago, these technologies didn't exist. So the new technologies actually cause a revolution in what we are able to do with diseases in the ataxia arena. Next slide. So these are some of the targets for ataxia drug development. These are all companies that have contacted National Ataxia Foundation. They have been in discussions with the National Ataxia Foundation about their goals, about potential funding, about ways they can help in drug development specifically for the ataxias. So with the companies looking at gene-directed therapies, there are two basic approaches. Gene replacement, where there's a gene that is underactive, as in Friedreich's ataxia, and would respond to being replaced by an active gene, and gene blocking, where in spinocerebellar ataxia, there's a gene that's producing an abnormal protein, which itself goes on to cause damage. And if we could block that gene, it would relieve some of the stress that's causing the disorder. Now, I'm hoping you will all have access to these slides so you can look at each of the individual companies on their websites, their research that they're doing, the pipelines that they've engaged, and also a little bit about the science behind how they're designing gene-directed therapies, either gene replacement or gene blocking. Next slide. In talking about the spinocerebellar ataxias, the genetic ataxias where there's a mutant or bad gene that is producing a mutant or bad protein, which may or may not do its normal job, but has toxic influence on the nervous system and specifically the cerebellum and its connections. So this little diagram starts at the top left with what's called a polyQ gene. That's a gene for ataxia type 1 or 2 or 3, where the mutation is an enlargement of part of the gene. The gene can be transcribed and create an abnormal protein, a polyQ protein. The protein is recognized as being abnormal and is broken down into fragments with an attempt of the nerve cell to get rid of it. However, the fragments fold up in an abnormal way. They clump together and make inclusions you could see at the bottom of the middle part of the slide. The small boxes that are arrayed along this pathway outline places we could intervene with drug treatments. And each of those boxes represents activities by one or more pharmaceutical companies or research labs to develop, for instance, at the very top, RNAi, which is an inhibitory chemical that prevents the gene from making the bad protein. So it's a gene blocker, as it were. Antisense moieties are also, the ASO moieties could also be placed in the RNAi box, doing basically the same thing, blocking the production of the abnormal protein. If the abnormal protein is produced, you can develop a drug that will prevent it from being chopped up into fragments, thereby sparing that buildup of the fragment inclusions. You could stabilize the fragment with another type of drug so that it doesn't become misfolded and turn into the aggregates and inclusions that we see further down the pipeline. Proteasomes are the parts of the cell that are responsible for getting rid of old or damaged proteins in the normal function of the cell. So proteasome-directed drugs could make it possible for these little garbage disposals, as it were, to be able to deal with those small abnormal fragments and get rid of them without getting clogged up. There could be drugs that would prevent the small fragments from clumping up, and there could be other drugs that could help get rid of the inclusions and aggregates if they happen to be formed. We know in the spinocerebellar ataxias that these abnormal fragments and aggregates and inclusions definitely upset and reverse normal cell functions. So anything we can do to relieve the burden of these abnormal fragments will help the cell function normally, make energy normally, go about its normal activities, and prevent the deterioration that these genetic changes can cause. Next slide. Epigenetic therapies are aimed at making a mutant gene behave like a normal gene. And there are 2, 3, maybe more companies that have been working on drugs that would, for instance, in Friedreich's ataxia, make the mutant gene produce protein anyway. You can see that the 3 companies on the slide with epigenetic approaches have chosen to begin with Friedreich's ataxia. But certainly epigenetic approaches could be applied to some of the other genetic ataxias as well. And the drug that BioMarin has been working with for a number of years now did work successfully in a mouse model of Huntington's disease, which is very similar to the mouse models of spinocerebellar ataxia. So we should keep our eye on some of the epigenetic therapies. Many of them come through high throughput screening, where known drugs, approved drugs, are being looked at in multiple test tubes to see if they can have any influence on a particular disease cell. So some of these drugs probably act epigenetically, forcing the mutant gene to behave like a normal gene. Next slide. Therapies directed at the abnormal protein that is being produced either in genetic ataxias or non-genetic ataxias like multiple system atrophy. The BioBlast company has had an intravenous trehalose, and many of you know about oral trehalose, which has been around and in discussion for a while. In their early studies with the intravenous trehalose, which is a form of sugar, they were able to stabilize the mutant protein, and it appeared that they could slow progression of, in that case, spinocerebellar ataxia type 3. This company was recently selling the IV trehalose protocol to another company called Selos. And this is not uncommon in the world of pharma, where they'll work on a particular drug development for a while and then may sell it or hand it off to another company that will see it further along the pipeline. Biogen working on an anti-alpha synuclein antibody to break up bad protein in Alzheimer's disease, in Parkinson's disease, and this could absolutely have application to multiple system atrophy, where a similar alpha synuclein protein is building up. Chondrial Therapeutics is doing a protein replacement therapy, and they've entered a phase 1 study now for Friedreich's ataxia, where because the problem in Friedreich's is a deficiency of normal protein, they have found a way to, by injection, replace it and get it back into the nervous system, the heart, and the rest of the body. And Horizon Pharma has some agents that have been around in the literature for a long time that seem to protect those protein fragments that we saw on the previous slide from clumping up and make it easier for the nerve cell to get rid of them. Next slide. I think the most interesting and most talked about area are drugs and vitamin supplements that are felt to be free radical scavengers, antioxidant, antioxidative stress-directed therapies. We know that anti-aging research is looking a lot at free radicals. Similarly, in a nerve cell that has been under stress from a genetic or non-genetic process, free radicals are produced. Mitochondria, which are responsible for making energy in the cells, become less effective at doing that. So why couldn't I just take high doses of vitamin E and slow up progression of my ataxia or prevent onset of my ataxia? Why couldn't I take high doses of vitamin E and live till 120? So these companies have looked seriously at the ability to use potent and directed antioxidant drugs to reach into the pathway of nerve degeneration in ataxia to see if it can slow progression or even turn the clock back. So here's a list of ones, some we've heard about, certainly in the Friedreich's ataxia world. The Reata and Retrotope companies have drugs that seem to stimulate mitochondrial function, protect the nerve cell from oxidative damage, and potentially slow up progression of Friedreich's ataxia. Any of these drugs, even if they begin testing in a particular disorder like Friedreich's ataxia, could be applied to any of the other ataxias where we suspect that there is a problem with oxidative stress and free radical damage. So even if a company starts out with Friedreich's, for instance, we are hoping in the future that many of these companies will also move ahead and test those hopefully successful agents in other genetic and non-genetic ataxias. Next slide. There are drugs in development that are focused directly on the Purkinje cell. The Purkinje cell is the primary nerve cell in the cerebellum, which coordinates signals going out for balance, coordination, suppressing tremor, and things like that. Currently, Biohaven is in a phase 3 study of their drug, which is similar to riluzole, the one that we mentioned before that's approved for Lou Gehrig's disease. They have a prodrug of it, which seems to improve Purkinje cell signaling, even if the Purkinje cell is under stress or partially damaged. It may also protect Purkinje cells from dying, or at least hold off that for a while. Cadent, which used to be called Ataxion, also has a drug that can improve the electrical firing of Purkinje cells, so they will function more normally, improving coordination and balance, even while the cell is under stress from genetic or potentially non-genetic factors. With the Purkinje cell-directed therapies, they could be applied to any form of ataxia where there are damaged Purkinje cells. And this could be non-genetic ataxias, it could be ataxias from stroke, ataxias from brain injury, any place you'd want to perk up the performance of a Purkinje cell, these drugs could actually be applied. Some of the other companies looking at agents directed at Purkinje cells, Steminent has a stem cell protocol that is being tested in ataxia type 2 and 3 and may come to the United States potentially later this year or next year. The stem cells that they're using may increase nerve growth factors, which are important for preserving and strengthening nerves and nerve cell pathways and connections. The other thing that can increase nerve growth factors, and we should not lose sight of this, is that exercise, rhythmic, repetitive, cardio-type exercise has been shown in the mouse model of ataxia type 1 to increase nerve growth factors. slow progression and prevent onset or delay onset of ataxia in those mice. We can't lose sight of the importance of all the non-drug approaches that we can use to help ourselves with ataxia. Next slide. Multiple system atrophy is one of the more common non-genetic ataxias. It is about 20 to 25% of the non-genetic ataxias that present after the age of 45 or 50. On the left side of this slide is a pipeline showing changes that occur in multiple system atrophy with buildup of, again, an abnormal protein, the damage that it causes, and how it leads to the symptoms of multiple system atrophy. On the right side of the slide, we have the disease-modifying approaches that are being looked at. The only company that has a multi-center drug trial going for multiple system atrophy currently is Biohaven, and they have a drug that fights inflammation. As you can see, it's third down on the list there, and may slow progression and improve symptoms of patients with MSA. Other approaches are being looked at single sites, single research groups, but anything that looks promising, as Dr. Shakkottai pointed out, could certainly move into a multi-center official drug trial. Next slide. This is, I think, a pipeline we've all seen from the FARA website. It shows what has been developed for Friedreich's ataxia, how each of the approaches, whether it's an approach to energy production and mitochondrial function, or gene replacement, or epigenetic therapies, how far along they are in the different phases. We hope soon, with the help of the National Ataxia Foundation, to have a similar pipeline beautifully displayed with wonderful graphics like FARA has been able to do here. And I'm hoping to work with the rest of our ataxia center directors and NAF at having a visible sign like this of how far we have progressed with the other ataxic disorders. Next slide. Companies with clinical trials for other forms of ataxia. Intrabio has a drug called acetyl-L-leucine, which is a derivative of a prior drug, acetyl-D-L-leucine, that was used in France for vertigo. It seems to have an impact on cerebellar Purkinje cells, so that it may actually be helpful for ataxia, not just vertigo or dizziness. And it is also felt to be neuroprotective and slow progression of ataxia in the animal models. Currently, it is being tested in ataxia telangiectasia, late onset Tay-Sachs disease, and Sandhoff's disease, but may well come into larger trials for the other ataxias. Eridel has a new technology where they can take red blood cells, erythrocytes, open them up in a black box machine, put whatever drug they want inside the red blood cells, and then seal up the red cells, put them back in the person who donated them, and deliver drugs that way. It is currently being used in ataxia telangiectasia to deliver dexamethasone, which is a steroid that is felt to have some symptomatic or disease-modifying benefit for children with AT. And certainly that protocol of using erythrocytes as little boxcars to deliver drugs at lower dose, potentially with fewer side effects, could be applied to some of the other drugs in development in the pipeline. Next slide. So with all these opportunities, how can we test them efficiently? We don't want to spend, 5 years working on one drug before we can move on to the next. We know we're dealing with, relatively small numbers of people with ataxia, that in order to run 10 trials at one time, we would probably run out of patients. So we have to think how we can make trials more efficient, and how we can do them faster so that all of these very appealing drugs can have their time in testing to prove themselves or not. Next slide. Roadblocks for clinical researchers and pharma developing these drugs, picking the right drugs, as Dr. Shakkottai pointed out, designing the trial properly. How many patients do you need? What are you going to measure? How long are you going to run the trial? Does it need to have a placebo? And how many sites you're going to have the drug tested at? You have to convince the FDA to let you move ahead. You have to get funding. You have to find sites that are willing to do the trials. You have to find subjects, and you have to do the work in a timely fashion. Next slide. So the tools that have made this possible and will be expanded and refined for continuing use have been supported by the work of the National Ataxia Foundation, the Friedreich's Ataxia Research Alliance, the NIH, understanding disease mechanisms, finding those drugs, getting a natural history baseline, making sure the rating scales that we use actually measure what we want to see. Biomarkers in blood, in spinal fluid, brain imaging can be helpful and have been developed. We have networks of clinical research centers under FARA and under NAF. There's a patient registry for Friedreich's ataxia and for the general ataxias, which can be accessed through the NAF website to help with patient recruitment. And we have databases to store all this data in for analysis. Future directions, which are being activated now, telemedicine by default. You know, a lot of us are doing regular clinic visits now by telemedicine, but there are several companies developing ways to do research visits via telemedicine as well. Those wearable devices, electronic monitoring, to monitor things in real time or at a distance, and international collaboration, which has begun now with ataxia type 1 and 3, as well as Friedreich's ataxia. Next slide. So these are the types of clinical trials which were described previously by Dr. Shakkottai. You can see what they require and how we have to make them work more quickly and more efficiently. Next slide. Funding is a public-private partnership. When we look at each stage of development, the phase 1, the phase 2, ultimately phase 3, to get to a phase 2 study could be upwards of $4 million for the drug that's moving forward. Now, when we're looking at $4 trillion, as we're hearing about on the news, 4 million seems like a small amount, but in order to raise it and to distribute and use those funds, it really is a partnership with government, private research foundations, pharma, and patient advocacy groups. Once we get to FDA approval, we need to continue to study these drugs over the long term. And this is something that we hope we'll begin to be doing within the next year or two, with hopefully our first approved drug for ataxia. Next slide. This is where you can get more information about clinical trials, the National Ataxia Foundation website, the Friedreich's Ataxia Research Alliance website, the Muscular Dystrophy Association website, clinicaltrials.gov, and the FDA website. Next slide. So thank you for your attention, and I believe we have time for questions.
Thank you, Drs. Perlman and Shakkottai, for all that outstanding information that you both shared today. We certainly are able to now begin answering questions that have been submitted prior to today's presentation and during today's presentation. You can still submit questions through your control panel. If you'd rather ask a question verbally, you can now start using your hand raise tool in your control panel, and we'll certainly do our best to answer as many questions as time allows. And Dr. Perlman, would you like to start us off with answering a few questions that you have?
A few questions did come into NAF prior to the webinar, and they're very, very good questions. One of them, quote, I am on 4-aminopyridine. It does help me walk. The rhythm of my gait is better, and that gives me more confidence to walk. Are there any studies showing which ataxias this drug works for? Now, 4-aminopyridine has been shown to be effective for episodic ataxia, where people have attacks of ataxia that then recede and then may come back later in the day or a few days later. And 4-aminopyridine seems to stabilize those attacks so they're fewer and less severe. They have been found to be helpful for persistent ataxia between attacks, and that is what this individual has noticed. So theoretically, if it's a symptomatic drug, it could be helpful with any form of ataxia, whether it's episodic or not. Currently, it is approved as a prescription drug called Ampyra or dalfampridine for multiple sclerosis. But since it became available as a generic, it seems to be easier to get by prescription for people who do not have MS, for instance, people with ataxia. And a companion question, is there a drug on the market now, or are they working on one that helps with spasticity, muscle tightness, but doesn't fog up the brain? And prior to the availability of 4-aminopyridine as a prescription drug for MS, it was and still is being formulated by compounding pharmacists for use for ataxia. But back in the day, when I was a trainee, it was in use for spasticity. Spasticity from MS, spasticity from spinal cord injury. So theoretically, 4-aminopyridine could be an anti-spasticity agent that wouldn't cloud the brain. And then one more question. What medicine can I try to slow the progression of spinocerebellar ataxia type 3? As I indicated in my slides, there is currently no approved or proven drug to slow progression of any of the ataxias, with the possible exception of a couple of the drugs in development for Friedreich's ataxia. However, the animal work done with the spinocerebellar ataxia type 1 mouse and the helpfulness of exercise, I think, is something that every ataxian needs to look at and make part of their daily regimen, with the hope that it will preserve function, improve function, and slow future worsening. Somebody had another question. Are there any treatments or clinical trials for idiopathic ataxia? Currently, there are no active trials in North America, in the United States, for non-genetic, non-multiple system ataxia. But as I indicated, any of the drugs under development as antioxidants, neuroprotective agents, or Purkinje cell-directed agents could be used in any of the ataxias, so we must keep our eye on that. One thing that came out of Europe a few years ago was a trial with riluzole, regular riluzole as it's approved for Lou Gehrig's disease, in groups of patients with ataxia, genetic ataxia, non-genetic ataxia, idiopathic ataxia. And there was some evidence that riluzole could help symptoms of any of these ataxias and potentially slow progression. So many doctors may decide to try regular riluzole for their ataxia patients independent of the cause. So why don't we go to some additional questions that Lori has.
All right, there's a question here. Are studies designed for early or advanced symptoms? And is one better than the other when it comes to starting a study?
Dr. Shakkottai might be able to answer that.
So I think it depends on, it goes back to some of the things that I mentioned regarding who's participating in the study and for what indication. So for example, an agent that improves symptoms would be potentially equally applicable to participants who have early stage disease versus later stage disease. The caveat, of course, is that we as Dr. Perlman mentioned, many of the symptomatic agents are to help the cerebellar Purkinje cells function better. So the later stage disease may not have as many Purkinje cells to work with, but I think symptomatic trials are more amenable to treatments that are at different stages of disease. Whereas a trial that's primarily meant for protection, we know from studies in other neurodegenerative disease, including Alzheimer's disease, that very late stage disease, at least in the context of a clinical trial, there may not be sufficient signal to look for slowing of progression, and early to mid-stage disease may be more amenable to treatment that slows progression.
Thank you, Dr. Shakkottai. We also have a question. Is it beneficial for two siblings experiencing the same symptoms to enter a study at the same time?
So there is no restriction on family members participating as long as each of them is separately informed and has an informed consent process. We do know that in particularly ataxias where the conditions are rare, it may be only possible to recruit multiple members in a family. The caveat here is we want to also know whether treatments are broadly applicable. So if siblings have more shared genetic information outside of even the mutation in their ataxia, so to have too many participants from a given family, for example, may potentially confound the interpretation of how broad the applicability of an agent is. But I think at least for the early stages, it would be very reasonable to have several family members participate as long as they are independently informed of the risks and benefits.
Great.
Can I make one additional comment about that? I know that in Huntington's disease studies, there was an interest in identifying participants, not by name, but by some numerical linkage, who were in the same family, anticipating that there's going to be other genetic factors that could influence the course of the disease or response to drug trials that might be unique to that family or group of families, other modifier genes. So I'm hoping that in the ataxia research world, we'll be able to look at some of these modifier genes that may run in families and, protect families from gene stress, et cetera.
Great. We're gonna take live questions. Stephanie Wilkins, do you have a question?
I do. Hi, Dr. Perlman.
Hi.
Do you know if any of the current clinical trials are on hold because of the COVID-19 virus?
There were two COVID-19 questions that came in. One was about the timeline for the various clinical trials, the gene blocker trials, the stem cell trials, and some of the others that we're hopefully going to be beginning later this year. Some of the companies have put their studies on hold until face-to-face visits can be started up again. Companies that are running trials now are continuing with the patients who are already enrolled, but are transitioning some of these visits to telemedicine visits until the live visits can be resumed, but not enrolling new patients. We're hoping that the pause for new studies and for ongoing studies is going to be lifted in a few months, and we're doing everything we can to keep all the foundation active for these studies. And then there was another question in apparently the last webinar. Ataxia patients were told that they were considered at risk for the coronavirus. My understanding is that we are still trying to figure out risk factors for who gets it, who gets most sick with it. But any ataxia patient who has weakness and incoordination that may alter breathing, certainly is at risk for more complications from the coronavirus. But because the majority of ataxia patients are not immunosuppressed, they shouldn't be at any greater risk in catching it. So that their risk is going to be the same as all of our risk. But should they have weakened breathing, decreased mobility, they would be at higher risk for complications.
I would want to add just one other thing. We know that one of the most significant effects for mortality from the coronavirus is age, and at least some of our ataxia participants, particularly sporadic ataxias, including multiple system atrophy, are at increased risk, potentially from what Dr. Perlman mentioned in terms of breathing, but also because of their age. And this is another thing that I think is important to consider.
Thank you. We also have a question: when developing a new drug, is evaluation a factor? For instance, if a person living at sea level takes a drug, could someone taking the same drug living a mile away or higher elevation have different side effects?
Dr. Shakkottai?
Yeah, I can answer that. In general, when we do the preclinical studies for things like pharmacokinetics, the pharmacodynamics, meaning how a medication's absorbed, how it distributes into the body, altitude doesn't conventionally play a role in the distribution of the drug or engagement of target. But there are environmental factors that definitely influence how somebody's takes up the drug. But I'm not sure that altitude would make a significant difference for at least the objective, measurable things for how a drug distributes and acts.
Great. Another question is, can antioxidant intervention potentially arrest progression of AAA repeat transcription expression? And how can one design a clinical trial for this given the inherently long time horizon expected to notice an effect or lack thereof?
Dr. Shakkottai, what do you know about the preclinical studies of antioxidants in animal models? And how could we design a trial in ataxia patients that wouldn't have to run for 5 years to show a benefit from an agent that may be mildly effective?
Right. So I think this is a very valid question. I mean, and this goes back to what the effect size is. And I'm going to I'm going to address kind of supplements as antioxidants and separate those from a study that was recently done in patients with Friedreich ataxia using omevaloxolone, which is a NRF2 activator. So that's not a conventional antioxidant, but increases the genes that are responsible for mitochondrial respiration. So that agent, at least based on the publicly available data in Friedreich ataxia, showed an effect size that showed not just slowing of progression, but improvement in symptoms. And I know that Dr. Perlman has more publicly available data on the study from having recruited participants into that trial. So I think what is important is antioxidants have a relatively good safety profile, and that supplements, for example, like coenzyme Q10, which may not have a large effect size when formally tested in clinical trials, may yet have some benefit in long term for as far as their safety profile goes. So I think so what I generally tell my patients is things like coenzyme Q10, which even if in a formal clinical trial don't show any benefit due to restrictions on the amount of duration of time, may have small effects that are still worthwhile over the long term. And since their safety profile is very good, it's a decision that an individual patient, based on things like affordability and other things, can take regarding antioxidants. Dr. Perlman, regarding...
They should certainly discuss any supplement or herbal preparation with their neurologist or primary care doctor, because there are unexpected side effects from some of these preparations. Certain herbs can affect blood clotting. They can affect the liver. They may seem harmless. They're sold over the counter at your local health food store, but I think you need to check with your doctor to make sure there's no interaction with other drugs you may be on and no other side effects that you could be sensitive to.
Great, thank you. We're going to, let's see, take a live question from Charles. Charles, you're...
Okay, I'm ready.
Unmuted.
Is there any research scheduled in the future for autosomal recessive types such as ARCA1?
I believe at least one of the companies working with genetic ataxia is planning on looking at some of the recessive ataxias, not Friedreich's, but some of the other less common ones. But there haven't been any protocols put out yet. I know that there's one or two investigator-initiated studies, for instance, of the Biohaven prodrug of Riluzole, which has been looking at a few people with recessive ataxia as well as some other rarer conditions. And pretty much working with any company that has a drug in the pipeline that has passed the phase 1 mark where its safety has been documented, your local neurologist could submit for a single subject trial of that drug to the FDA with the cooperation of the company that's developing it. So, I think these are things that, could be looked at on an individual level, even while these larger multi-site studies are ongoing.
Thank you.
All right. There is a question here that says, I was diagnosed with SCA1 about 8 years ago and have symptoms, so I'm pretty sure I have ataxia. My question is regarding the number 1. Do I need to recheck about the number, or is it very much determined at that first check?
What was the question exactly?
So this person was diagnosed with SCA1 about 8 years ago. Their question is, do they need to recheck that diagnosis at this time, or is that pretty solid that diagnosis is correct?
Genetic testing in the last 10 years has been pretty reliable. I think one thing that comes up rarely is an individual or a family tree can actually harbor 2 ataxia genes. I have seen families that have ataxia type 1 plus ataxia type 8, ataxia type 6 plus ataxia type 8. So if the original testing was done only for that gene, and there's any concern that the course is unusual, or perhaps there's another genetic factor involved, there could be a recheck for a broader range of genes. But it sounds like the mutation for the ataxia type 1 gene is pretty convincing and doesn't have to be repeated.
And I would just add just one other thing. I mean, I think if I'm interpreting the question correctly, the person is asking if their number, meaning SCA1, will change over time. And I just want to say that the numbers SCA1, 2, 3 are based on the gene that harbors the mutation. So someone who harbors the mutation, and the reliability of the test is now close to 100% based on the PCR analysis, that number does not change. So someone with SCA1 who has a mutation in the ataxin 1 gene will continue to harbor that mutation. and that number will not change.
Good point. Another question is, do you know on the SCA1 trial how the drug would be given to the patient?
Are we referring to the Biohaven study of
I
their
believe so.
prodrug of riluzole?
I believe so.
Because that was an oral drug. Now, some of the gene-directed therapies, for instance, the gene blockers, which could absolutely be applied to ataxia type 1, are given by injection into the spinal fluid or potentially directly into the brain. So that's a very different approach. But the current drug trials are oral agents, both for the genetic ataxias and multiple system ataxia.
Gay Manning, do you have a question?
Yes, I wanted to know what the fatigue, what causes with the ataxia. I seem to be fine for a while and I can walk, but then I get really tired quickly. I have had lung cancer where a small half of my upper right lobe had to be removed. I don't know if that is the ataxia or is it the result of the lung operation.
Do you have a diagnosis of a genetic ataxia?
It says that it was a genetic, but it hasn't been totally proven. I do have an appointment at the Pixel Clinic in Gainesville. Is that one of the ataxia centers?
Yes, there is an ataxia center at the University of Florida at Gainesville.
Right, I'm going to be going there. They had to reschedule my appointment until June 9th. But I'm just, as I hear you talking, they said it was hereditary spinobello, cerebellum ataxia, but they didn't really do the final test to find out if it was hereditary. And I'm the only person that I know of in my family that has it, if it is hereditary. And therefore,
symptoms come on before the age of 50.
Pardon?
If you have symptoms of ataxia that came on before the age of 50, it increases the likelihood that it's potentially genetic. If you're over the age of 60 when your symptoms began, a genetic factor is less likely. But you're right, you need to be examined and have the testing done. However, symptoms of ataxia.
My ataxia did not. I've had the brain scans and I was, it said that I have the atrophy on my brain that has to do with the gait and the balance. However, it didn't occur until I was about 75. Okay. So I would say that it's probably not hereditary. Would you agree with that?
Is the chance. About 10% of the people I see over the age of 60 with no family history do end up having an ataxia gene. So it is a possibility, but certainly if you're a lung cancer survivor, there could be other factors contributing. There could be autoimmune factors. Your immune system could have developed a sensitivity, which is putting stress on your cerebellum, as you pointed out. And certainly the episodic features, the good days, the bad days, the fatigue, can interact to make symptoms worse or to make them better for a period of time, whether it's genetic or non-genetic. So definitely follow up with the doctors in Gainesville and see if you can get a better answer.
Okay, thank you very much.
One other thing about the age and the lack of, or whether it's a genetic or non-genetic form of ataxia. We do recognize now that what we used to think of as a sporadic adult onset ataxia, which is more an idiopathic ataxia of late onset, many of them are due to mutations in genes, not not the polyglutamine expansions, but other mutations in genes that do cause late onset genetic forms of ataxia. And I do want to point out one recently identified ataxia that's actually relatively common and late onset, which is a repeat expansion in a gene known as RFC1, which is a recessive ataxia that's most directly associated with a condition known as canvas syndrome, which is cerebellar ataxia with vestibulopathy and neuropathy. But in a substantial number of patients, and we have done a lot more testing in our later onset ataxia patients and have identified mutations in this gene that potentially make it as common as Friedreich ataxia is in younger patients.
Great. So we're going to take one final question here. We have a question that was submitted. I noticed that at least one SCA2 trials are in phase 2. How soon will it be in phase 3?
Which trial was that? Because
Does
there are a
not
couple
say.
of trials that have included ataxia type 2 that have been in phase 2. One is the stem cell study in Taiwan, and the other is the Biohaven study, which has moved into a phase 3 now, which includes ataxia type 2 patients.
Maybe we could comment on what your thoughts are on where those are in phase 2 and how long it usually takes to advance into that phase 3.
Well, Dr. Shakkottai may want to address that.
So I guess the typical timeline, it's hard to, as Dr. Perlman mentioned, it's hard to know which specific study that's being referred to. But typically the timeline of a study moving from phase 2 to phase 3 really depends on whether the endpoints were met in the phase 2 study. It typically takes a few years, between 2 and 5 years, to conduct a phase 2 study, from the planning to the completion of all the analysis to see if the study endpoints were met. For example, the omaveloxolone study for Friedreich ataxia fairly rapidly transitioned from a phase 2 to a phase 3 study. In fact, there was some overlap between the phase 2 and the phase 3 studies based on some interim analysis. So it really depends on whether the endpoints, both for safety and a phase 2 study is not powered, meaning doesn't usually have enough participants to know whether it's effective or not. But if sometimes the effectiveness is more than originally planned, a phase 2 study can very quickly transition to a phase 3 study.
And some studies are designed to be a combination phase 2-3 if they're dealing with, an orphan drug for a rare disease, to avoid having to design and pay for two, kind of larger studies, a 2 and a 3. They will sometimes combine them as a phase 2-3 study.
Great point. Thank you again, Dr. Perlman and Shakkottai, for those great answers and great information. Just wanted to remind everybody again that more information on clinical trials and studies that are recruiting are available on NAF's website. Also, just wanted to put another reminder out there. If you've not already done so, please consider joining the CORDS registry. This registry is a really important recruitment tool for ataxia research studies and clinical trials, and that is also available on NAF's website. Also wanted to let you all know that a recording of today's webinar will be made available, and I know we didn't get to all of the questions that wanted to be asked or were submitted, so you can send those to naf@ataxia.org. And thank you all again to those of you who were able to join us today for today's webinar. We truly appreciate it. And hopefully we'll see you at our future webinars. And please take a moment to complete the post-webinar survey. Enjoy the rest of your day. Thank you.